As we can see here, this is the beginning of transcription and the beginning, therefore, of protein synthesis. Here we have, of course, the genes that will be transcribed. We have the promoter, which is going to basically allow the RNA polymerase to begin the transcription. Without this, nothing's going to happen. Of course, we have the small RNA subunit, the large RNA subunit. These will combine, they sort of engulf the promoter, which basically starts the process of transcription. We have the repressor and the regulator. Now, the regulator will control how frequently, how often basically, how many times it's produced and what is produced exactly. And as you can see, the repressor and the regulator fit into each other. So in certain cases, depending on what is produced exactly in this combination, the repressor will cover the regulator, meaning the genes will not be able to be produced or transcribed. And this is based on the regulator and the repressor. They work together to basically regulate or repress. We also have obviously the genes themselves that will be producing the protein and therefore the end product. And that's about it. Okay, in this picture, we can see that the RNA polymerase, the small and the large subunits have combined across the DNA and are beginning their job, which is basically to unravel the strands of DNA. As you can see here, double helix, continuation of double helix, and here is the section that is going to be transcribed. So what basically happens is it is separated from the anti-strand so that it can be coded for. Of course, the coding strand will produce this mRNA. And for example, if this base here is A, then basically the thing that will come out of the mRNA will be U. As the base is, remembering that RNA has, instead of T, it has uracil, thymine uracil, which means that for A you'll get U and CG and the same. So basically, even though you do use the coding strand, what you're effectively getting is the anti-strand, because of course you can't code directly for DNA, you can only code for its, what would you call that, its opposite strand basically. I have to find the word for that and put it in a later video. Anyway, that is effectively what's happening. Of course, the regulators have indicated that now is the time to transcribe some DNA, some genes of some sort to produce a protein, and this section therefore is unraveled, producing of the same length and of course opposite bases mRNA. Regulators are very important in this entire process, and indeed many processes within the human body. They control the genes themselves, they control their expression, their inhibition, but they also control, in fact, the splicing or the spliceosomes and where and how they are associated, which is extremely important. So the regulators, in fact, are even more important in some ways than the genes themselves, because the genes themselves only code for proteins, whereas the regulators can change how the proteins interact, how the amino acids themselves interact, and also the alternative splicing, a very big idea as we've just discussed. So the regulators enhances, silences, and etc. Enhances will increase the likelihood and indeed the transcription rate of certain genes, where silencers do the opposite, they inhibit the transcription rate of certain genes. All very, 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 very important. Here we have the idea of alternative splicing. This is where, following the formation of mRNA, as we talked about previously, the splicing enzymes are placed within the DNA strands and remove certain introns, which are non-coding parts of DNA. Promoters, repressors, regulators, they're all things like this. They control how often how many and what to produce from the genes. What effectively happens is a spliceosome is formed from these splicing enzymes and cuts out the introns. But why we call it alternative is because different introns can be cut out, giving you multiple combinations. And this, in combination with translation, would explain why so many proteins are able to be produced from a limited number of genes and amino acids. And indeed, this is the bridge between premature mRNA, and after this is complete with the removal of certain introns, we get mature mRNA. If you can read that, it says mature mRNA, which is what we have now. Now we have mRNA, but we need to translate it, which is why the process is called translation, the second process in protein synthesis, to an amino acid sequence via tRNA. What happens is the ribosomes read the mRNA sequence and begin forming an amino acid chain. It always starts with the codon A, U, G, which is, the mRNA strand or tRNA strand is AUG, which means that the complementary strand must be, there's that word I was talking about, I found it, must be TAC. That is TAC. But it's RNA, so it's AC. Okay. Of course, the amino acids are then collected, and when they form a big enough chain, it becomes a polypeptide. Of course, the amino acids are bonded via peptide bonds, polypeptides, because there's more than one. And when you have a long enough chain, this will combine with other polypeptide chains to form a protein, hence the protein synthesis, because you have now synthesized protein. But now we must look at the minutiae of this process. Transcription occurs in the 5' to 3' direction, meaning that when mRNA is produced from the coding strand, it is produced in the 5' to 3' direction, which is important to know for knowledge's sake. In tRNA, there are anticodons. These are 3 bases. So, for example, the start base, as I said, would be AUG. That's an anticodon, that's the starting anticodon. These attach to the mRNA's codons, codon, anticodon. These are very different to the triplets that are located in DNA. But sorry about that. But effectively, they allow for multiple amino acids, and therefore combined with alternative splicing and amino acid chains, many proteins. We can see here that tRNA has a cloverleaf shape, which allows it to transition various bases and amino acids into peptide chains effectively in the right place. These are made up of single strands of RNA, yes. So this would be, for instance, AUG, and then this would maybe be UCA. It doesn't matter. But what I'm saying is this is all an RNA strand placed in such a shape to allow for convenient and efficient allocation of amino acids into a peptide chain. The 3-letter codons in mRNA translate to complementary anticodons in tRNA, and each of these codes in tandem with each other to a specific amino acid, which in turn can be placed in the chain and, depending in their orientation, can produce a different protein. As we have previously talked about, there is the AUG, which is the start sequence for the transcription and translation process, and this basically starts the entire process of producing the amino acid. UGA is in fact is one of the codons that is a stop codon. It stops. It tells the anatomy that it's finished, the sequence is finished. Beginning of sequence, end of sequence. AUG is the single start sequence. However, UGA, there are two other codons that will do the same function of stopping it. We now move to protein structure, as you can see up here. We have the first structure, which is an amino acid chain. So this is when they're connected by peptide bonds, giving their name polypeptide. After this, the positive and negative parts, because there are 20 amino acids. And of these amino acids, some are positive, as in the R group is positive, but we'll have to refer to that later. Some are negative and some are polar. When you have the positive, negative, and the polar interacting, you can either form a helix shape where it twists around an alpha helix, or if there are consecutive positive and negatives in such a grid manner, it can form beta pleated sheets, which is, and this is the fibrous form of protein. We move on to the third structure where it becomes globular. This is where one or both of the structures fold in on itself because of the R groups interacting to such a degree that they do not form a concise and strong structure. They instead form globular structures. The fourth, also globular, is where one or more of the globular individual pieces comes together, connected usually in the middle by an ion of some sort, not iron Fe, but an ion, as in a charged atom. And these are the four structures of proteins. That really doesn't have much in terms of fibrous or globular. This is fibrous, globular, and of course globular.